Great. So our next short talk is by Dr. Aman Sharma. He's the CEO and founder of ExoCan Healthcare Private Limited. Aman started his company in 2015 soon after his PhD in biotechnology on high-grade brain tumor glioblastoma. His company focuses on developing cancer diagnostics and drug delivery technologies based on exosomes, and his talk will also be about targeted drug delivery using exosomes today. Welcome, Aman.
Okay, good morning everyone. Acknowledgements first. So I would like to thank WRCB and BIRAC for funding support and Venture Center for NCL Innovation Park Pune for providing incubation facilities. I request all of you to please raise your hand if you have heard of the word exosomes. How many of you? Wow, that's wonderful. I'm really happy. That was not the case when I started this company in 2015. There were probably I think we from India we had less than 10 paper in PubMed, and few of them were reviews actually. So technically, India was not aware of exosomes, and at the same time we were having thousands of researchers, doing research in the area of exosomes, starting from cancer, diabetes, and whatnot. So let me show you few of the science and the business aspect of how I turned from a scientist or researcher to an entrepreneur. And this is a famous, if you guys have heard of Tesla Tower. Nikola Tesla tried to build a tower which could actually generate or transmit energy without any wires. So it was a wireless communication, but the tower got failed because JP Morgan stopped the funding. So funding in science is equally important than, I would say, in fact, science. So what are exosomes? So as most of you people know, so I can say, you have, a typical email, you have a sender, receiver, and some encrypted message here. So there are two cells basically, for example, recipient and receiver cell, sender cells, and there is a communication that is happening via exosomes. So what we do as a company and how we took this challenging task and what sort of technologies we established. I'm going to talk. Some of the data I'm not going to show here because of the confidentiality, but I can discuss anytime off the record if any one of you is interested. So this is a typical image of exosome communication between cells, and there are basically two types of communication which can happen through exosome. I would say rather mode of transmission. One is receptor ligand mediated uptake. It's pretty specific, and then you have phagocytic system, which is random uptake or exchange of information between two cell types. So this is the basis of any type of targeted exosome drug delivery, because you want to make sure that whatever you're sending in exosome is actually going to the right cell or cell of interest. So I'll talk more on this particular systems and how we approach this. So basically, you name any kind of targeted drug delivery across diseases, from CRISPR to antibiotics and whatnot. You need to have a delivery system basically. We are using at the moment lipid nanoparticle derived system, which is sort of being outdated. So for example, you have a target cell here, any kind of cell, and it is expressing a certain receptor. For example, here you know what is the receptor, and you have a particular delivery system. It is at the moment because exosome research is nothing is approved by FDA at the moment. Everything is in R&D pipeline or some different phases. So you have these exosomes, and you engineer these exosomes, this is a simple cartoon. you have some exosome surface protein and you have a targeted ligand which is cognate to this receptor. So you put this construct in a cell of production, which is, let's say, here, and then you harvest exosomes, isolate, you load your drug. So your basically exosomes are engineered exosome which will now can only interact with this particular cell type. You load your drug, purify further, and ready to use, go to mouse or humans, if so, whatever. So what are the problems here and how did we solve? So there are two basic problems in this particular system. The biggest problem is here. The isolation itself is pretty challenging. I can proudly say ExoCan developed world's fastest and most efficient exosome enrichment and isolation technology, and we have been helping Indian community for last 5-7 years with this, and now you see a lot of exosome researchers are coming up. So we first established this isolation technology, then we established some other cargo analysis technologies, and I'm going to discuss two of these technologies here. One is drug loading, because once you load the drug here, you engineer the exosome this way. Now you need to load the drug. You can engineer exosome. It's any simple molecular biology lab can engineer exosomes. Order the clones and just transfect the cells and get these exosomes. The real crux is here. You want to put your siRNA, you want to put your peptide, for example, encapsulated peptide. You want to put your small molecule and whatnot. There is no optimized technology or standardized systems, and most of the lab across the globe are just on this particular hunting space. Everyone is using different strategies to engineer exosomes, and this particular field is a gray area, I would say, because there is not much of innovation. So we tapped here. So as I mentioned, there were two problems, and I would say they still persist: rapid isolation and pre and post drug loading. Pre and post drug loading isolation, plus efficacy of drug loading, because you want to load high amount of drug in the engineered exosomes if you want to make a targeted drug delivery into reality. What are the methods as of now for drug loading? There are typical non-scalable lab-based methods, sonication, electroporation, detergent methods, and a typical problems they are associated. They would show aggregation, membrane disruption of exosomes post these methods. Poor efficiency, because you can't load beyond a certain amount because of the, let's say, for example, this exosomes are naturally very, it's like a nature's concrete, I would say, because the lipid raft is so strong that even detergent would not be able to lie. So you would be surprised to know that even in the gut pH of 1, exosomes are pretty much stable, and that is why they are now being used as a they are now being seen as a novel drug delivery method, because they can bypass the liver system and all because of more bioavailability and etc. So this particular structure, lipid bilayer is, and that lipid raft structure is pretty tight. So you can't load beyond a specific quantity or of that particular drug. And the biggest problem, if you want to translate these all findings, if you even reach to that stage, so far so good, everything is okay, there is no problems here, but the batch consistency. Because if you want to go to the patient or preclinical studies, you need to make batch consistency. So the few slides I will show how we developed drug loading strategy and what we could achieve, and some other details. If someone is interested, I am ready to talk off the record, because this is being recorded, so I will talk, I'll be cautious here what I am going to talk and what not. So this is a mesenchymal stem cell. We use the mesenchymal derived exosomes. This is a typical FACS characterization using beads, whole micrometer beads, and then we did a characterization, typical couple of markers. So I'm showing not all of the data, representative data, then we did anti-A to check the size. And so this is the first, we characterized the exosomes from MSC, and then we loaded drug in these exosomes. And what we could achieve, we used the surface modification strategy to make sure that we are able to load high amount of drug in these exosomes. There must not be any aggregation post-drug loading, because that is the prerequisite to increase or sustain or maintain the bioavailability of exosome once they are in a preclinical or and serum system or in vivo system, I would say. And there must be functionality of these exosomes must be retained, and batch stability must be there. So if you see here a typical image after sonication, electroporation and our approach, so we have a product with which you can actually get a visible palette of exosome, which is not possible with any other global method or product. So this is the typical, we use 3 different approaches, including ours, we loaded the drug and paletted exosome. So I'll show in few more slides later on how we purified and all. And then we see that we are able to load up to 47.4% of drug, which compared to conventional method is just pretty high, I would say, just very less. Now the second point I mentioned, there must not be aggregation of exosomes. So before and after loading, we did SEM to check if their particles are monodispersed. And if you see in SEM, they are monodispersed, and this one particular concentration combination we have shown here. Lipid raft structure must not be disturbed. If it is disturbed, then they are functionally not viable, and the whole concept is useless. So we did a flotilin 2 is a lipid raft associated marker, conventional exosome marker. We did a blot, and pre and post loading, there was no differences or leaching out of this protein, making sure that these particles are having retained functionality of lipid raft structure. Then this is a simple in vitro assay we did for this particular drug I'm showing here for cisplatin. So if you see there is IC50 remains more or less same even after keeping. So we load the drug, we store the exosomes at -80 freeze thaw cycles, and there's some more data. I'm just showing representative data. Even after 5 days, the IC50 remain more or less same. There is no drastic difference, confirming that there is a batch stability of these formulations. Now you have loaded the drug in exosomes, and now you want to, so you have a cell system, you isolate exosomes, then you load your drug, and then you purify, and then you again use it for any downstream purpose. So at the moment there is in the across this particular field, you have no other method than this ultra sucrose gradient to purify exosomes. There is no other method. What I'm going to show here is that it's a 10-minute process, simple 10-minute benchtop centrifuge, and you get a layer separation. Like you have a 3-part phase separation here. This middle layer will have, fluffy layer will have impurities, all the impurities, and this upper layer will have a transparent layer. It will have purified exosome. In 10 minutes, no expensive machine, nothing, straightforward. So if you see so we have a technology to isolate exosome within, you can isolate in 20 minutes, followed by drug loading, let's say an hour, maximum an hour, if someone is not experienced, and for the 10 minutes of purification. This is the world's most fastest and efficient workflow of exosome research. We have recently got US patent also, so we have patents for few of these technology in India, US and China. So if you see, after a purification step, there is more data, but I'm just showing SEM data. So after purification, you see there is in the SEM, the exosome particles you can see here. So most of the impurities, like we could actually precipitate 98, I would say 99% of impurities. These are two different conditions. So we could get rid of most of the impurities which are non-exosomal from our exosome sample. And when we, that fluffy layer, the intermediate layer, when we did SEM of that, we did not see any exosome particle in the background, only these were proteinaceous crystals, making sure that we got rid of any kind of impurity present in the exosome sample. These are the capabilities that we have developed in last 5-7 years, from purification, cargo loading, enrichment, analysis, and whatnot. So we are also into engineering. So I think this is it, and I would be happy to take any questions. And of course, exosome can solve any kind of medicine problems in next 5-10 years. Thank you. Thank you.
Hi Aman, nice talk. So I have two very nice questions. The first one is, where do these exosomes really have a higher edge over, say, biocompatible nanoparticles for drug delivery? And the second one is, the cells inherently have exosomes. So, and in cases of a disease condition, they can sometimes have a double-edged sword kind of a function, where they are sometimes kind of leading to the progression of the disease also. So in that case, if we are using exosomes for drug delivery, will there be any kind of interference from the cell's inherent exosomes?
So nanoparticle system is it could be immunogenic. There is a lot of coronation which can happen once you put in the delivery, the involve bloodstream and all. Exosome being natural, your drug or molecule of interest can ooze out from nanoparticle no matter how strongly you have put that, used chemistry to put that in. But in case of exosomes, their lipid raft is like, as I told, it's cement. Nothing can, if you package it well, it will not come out. And second question was, what was that? Sorry. So for that you have to basically choose what production system you are using for isolation of exosome, engineering of exosome and drug loading, because if you're using a normal cell derived exosomes primary, they will not be like cancerous, or of course they will contain some microRNA proteins and DNA internally, but it's like transitory effect. So if microRNA goes into the cells, if it is not something like oncogene or something, it will just be cleaved within few hours, metabolized in the cell. It will not have any major impact on the recipient cell.
Thank you. So we are one talk away from a break, so any questions you can post to Aman during the break. So we're trying